Welcome back to MicroVerse. We have now completed bacterial taxonomy, where we learn how bacteria are classified, identified, and named. So, before studying the internal structure of a bacterial cell wall, we need to begin with something we can observe directly. What does a bacterium look like? Bacteria occur in different shapes, and they may also occur in characteristics arrangement. So, today's question is what determine the shape and arrangement of a bacteria? So let's understand both concepts together. The shape of bacterial cell is mainly determined by its cellular structure and genetic characteristics. The major bacterial forms include cocci, bacilli, vibrio, spirilla, and spirochetes. So let's look at them briefly. Cocci are spherical or approximately round bacteria. Examples include Staphylococcus and Streptococcus. Bacilli are rod-shaped bacteria. Examples include E. coli and Bacillus species. Then Vibrios. Vibrios are curve or comma-shaped rods. The classical example is Vibrio cholera. The next, Spirilla. Spirilla are rigid, spiral-shaped bacteria. Then the spirochetes. Spirochetes are thin, flexible, spiral-shaped organism. An important example is Treponema palladium, the causative agent of syphilis. Some bacteria may also show filamentous or pleomorphic forms, meaning their appearance may vary depending on the organism and conditions. So, first concept is simple. Shape describes the form of an individual bacterial cell. The next, arrangement of a bacterium. Now comes the second concept, arrangement. Two bacteria may have the same basic shape but appear differently because of the way the cells divide and attached after division. For example, cocci may occur individually in pairs, chains, clusters, group of four or packets. This gives us characteristics arrangement. So first, coccal arrangement. In this, first one is a diplococci. When cocci occur in pair, they are called diplococci. A clinically important example is Neisseria gonorrhea. Then the streptococci. When cocci remain attached in a chain, they are called streptococci. Example is Streptococcus pyogen. Then the staphylococci. When cocci occur in irregular grape-like clusters, they are called staphylococci. The classical example is Staphylococcus aureus. Then tetrads. Some cocci occur in group of four known as tetrads. Then sarcine. When cocci form characteristic cubical packets, they are called sarcine. Then next one, bacillary arrangement. So bacilli can also show characteristics arrangement. Two rods together are called diplococci. When the rod remain attached in chains, they are called streptobacilli. Some bacilli may also appear arranged side by side, producing a palisade. So why does arrangement matter? At this point, you may wonder, you should we care about the arrangement? Why should we care about the arrangement of bacteria? Because shape and arrangement provide important morphological clues during laboratory identification. When we examine a stained bacterial smear under the microscope, we don't see the name of the organism. We see its shape, arrangement, staining characteristics, and other features. This observation helps the microbiologist narrow down the possibilities. For example, gram-positive cocci clusters immediately suggest a staphylococcal pattern. Then the gram-positive cocci chain suggests a streptococcal pattern. So morphology provides an important first clue, although morphology alone is usually not sufficient or definitive identification. Then we go for shape versus arrangement. So let's make the distinction very clear. Shape means what does one bacterial cell look like? Coccus, bacillus, vibrio, spiral, and so on. Then arrangement means how are the cells positioned relative to one another. Pairs, chains, clusters, tetrads, packets, and palisades. So, shape describes the individual cell and arrangement describes the group. That distinction is very useful in examination and in practical microbiology. So, if asked what is the difference between bacterial shape and arrangement, answer is shape refers to the form of an individual bacterial cell, whereas arrangement refers to the pattern formed by bacterial cells after cell division and their relationship to one another. And remember this Staphylococcus seen in clusters, Streptococcus in chain, Neisseria in pairs, Vibrio cholera in comma shape, then Treponema pallidum in spirochetes. So here I give a memory trick. Shape means single bacteria and arrangement means social. So think shape what one cell look like and arrangement said how the cells stay together. That's all you need to remember the distinction. So today's question was what determines the shape and arrangement of a bacteria? Bacteria occurs in characteristic forms such as Coca, Bacilli, Vibrio, Spirilla, and Spirochetes. Their cells may then form characteristic arrangements such as pair, chains, clusters, tetrads, packets, and palisades. Together, these morphological features provide important clues for bacterial identification. So now that we know what bacteria look like, the next question is what gives a bacterial cell its shape and structural strength? That brings us to one of the most important structure in a bacterial morphology, the bacterial cell. In the next lecture, we will understand its structure, composition, and functions. So thank you for the learning with microverse. Keep learning, keep growing, and let's master microbiology. One concept at a time.